Good morning, everyone. I'm glad to introduce Dr. Valeria Lencioni as keynote speaker for the Midterm Conference within the European boDEREC-CE project. She's an hydrobiologist with experience on invertebrate taxonomy and stream ecology. She has 2 decades of experience in coordinating work packages or projects within national and international frameworks. She is currently working as Chief Curator of the Invertebrate Zoology and Hydrobiology Department at the Science Museum of Trento in Italy. We are aware of her experience in the field of emerging contaminants and her contribution to this topic, which is gaining increasing attention in recent years. She participated as project coordinator in the project RAISE-TN, Environmental Risk Assessment of Emerging Contaminants in Trentino Rivers, Effects on Wildlife and Human Health, aiming at evaluating the danger and toxicity of emerging contaminants for the aquatic wildlife and humans. For this reason, we believe her voice is a crucial addition to the conference. I would like now to welcome Dr. Valeria Lencioni to the conference and thank her for her important contribution. I will now leave the stage to Valeria.
Thank you very much, Francesca, for this very nice and perfect introduction. I am Valeria Lencioni. As Francesca told you, I am an hydrobiologist, and my ecological research focuses on the effects of climate change and pollution on biodiversity in fresh waters, mainly benthic macroinvertebrates in mountain streams. I don't know how many of you are familiar with these small animals. Small, but you can see them with naked eye. They are moving on and in the substrate of all freshwater habitats, from ponds, lakes to streams, springs and rivers. And most of them are represented by insects, the juvenile stages, the larvae and Eupholometabolus pupae also, but not the adults, because save the case of some beetles, the adults are terrestrial. And then crustaceans, mollusks, trichlads, annelids, acari, and nematodes. My talk will focus today on the environmental risk assessment of emerging contaminants in Trentino rivers, the RAISE-TN project funded by the private foundation Calitro. 5 research work packages and 1 devoted to the dissemination and sensibilization. And my talk will focus on the main results and conclusions on 2 of the research work packages focused on chemical pollution and effects on macroinvertebrates, and of course also on the dissemination and sensibilization work package. The environmental issue within which this project was conceived is the contamination of fresh water, specifically rivers and mountain streams, by contaminants of emerging concern or emerging contaminants. New compounds whose presence in nature has not been recorded before, contaminants that for a long time have not been considered as biohazards, and all contaminants that are now becoming problematic. Each one of these compounds is generally present in freshwaters in trace amounts, but the problem is that in freshwaters we have a cocktail and complex mixtures of hundreds up to thousands of active ingredients. Many are not regulated by law, and so in fact they are not monitored. The list is quite long of emerging contaminants. We consider active pharmaceutical ingredients, illicit drugs, personal care product actives, newer pesticides, endocrine disruptors, nanomaterials, and microplastics. There is a constant need for more data concerning these compounds to define the regulation of their use and establish solutions for their management, removal, and mitigation of their toxic effects. Regarding pharmaceuticals and personal care products, the main source to fresh waters is represented by the effluents of the wastewater treatment plants, at least in urbanized areas. But these problems regards more and more the remote areas, so high altitude water bodies, where these compounds are carried at medium-high distance from the source by the wind due to the drift effect. They are trapped in glaciers and released in the melting water. So we found in glacier-fed streams pesticides, some of old generation like chlorpyrifos, and then synthetic fragrances like tonalide and microplastics. Entering the ecosystem, they enter the trophic change and are bioaccumulated with biomagnification processes whose effects are still little known at ecosystem level. Our case study within the RAISE-DM project is located in the Italian Alps, in the Trentino province, so in northeast Italy. It's the Noce River and its main high mountain tributaries, the Vermigliana stream and the Presena stream. We selected 4 sites. The uppermost one just in front of the Presena glacier, and then 2 sites located in the Vermigliana stream, one upstream and the other one downstream of the wastewater treatment plant located in the Tonale Pass. And then the last one located 13 kilometers downstream of the Campodeno wastewater treatment plant in the Mezzolombardo municipality, few kilometers upstream of the confluence between the Noce and Adige River. Several stress factors are acting and pressing this catchment, from hydroelectric power production to the intensive mountain agriculture and tourism. Regarding tourism, it is characterized by the strong seasonality due to the presence in the uppermost part of the catchment of famous ski resorts like the Tonale village, and the Presena glacier too itself is exploited as a ski slope. So we have a strong seasonality for tourism here. The village is very crowded in winter, and as a consequence, a strong contamination, so higher level of contamination in winter in the streams, specifically in the Vermigliana stream. Despite the high pharmaceuticals and other emerging contaminants removal rate that we detected for both plants in Tonale and Campodeno, their effluents and of course the stream which is downstream of the plants resulted contaminated by a plethora of 80 pharmaceuticals and 25 personal care products, with the highest level of contamination in winter. As we can see in this graph, I took it as an example, is the site located in the Vermigliana stream, only 50 meters downstream of the plant, located in the Tonale Pass. We see the contamination in winter in blue, in summer in red of this site. In winter, due mainly to the pharmaceuticals, paracetamol and ibuprofen, and the personal care products, the artificial sweetened sucralose and the synthetic fragrance galaxolide. In summer, the most concentrated contaminants were again the sucralose and the galaxolide, besides other pharmaceuticals like valsartan, diclofenac, and ketoprofen. We classify the risk, the environmental risk of these emerging contaminants, and our analysis highlighted as antibiotics like amoxicillin and clarithromycin, and anti-inflammatories like ibuprofen and diclofenac are at high risk for the environment, based on their measured in pink or predicted in light blue environmental concentration. It is interesting, for example, the case of ibuprofen that resulted at high risk for the environment, only considering the winter concentration measured in the Vermigliana stream. So overall, this analysis highlighted that there is a potential risk to aquatic organisms, particularly in winter. And we evaluated this risk to wildlife. at different levels of biological organization, from gene to ecosystem. Our results emphasize the negative effect of the plants on wild biodiversity at the different levels of biological organization. Starting from the community level, we observe the local extinction of species at times and sites of maximum contamination. So for example, during the ski season in the Vermiliana stream, downstream of the plant, we didn't collect several taxa, I listed here, that were present in the same period, at the same date, just 100 meters upstream in the same stream, so upstream of the plant. Among these, some by indicators of metal and in general chemical contamination, and also by indicators of eutrophication. In fact, another negative effect of the presence of the effluent is the enrichment in nitrogen and phosphorus, so creating this condition of eutrophication. In summer, so just when the contamination was minimum, the community living upstream and downstream in the Vermiliana stream of the plant resulted more similar to each other. So we highlighted there's still a high capacity of recovery of the ecosystem in this condition of strong seasonality of the stressors, besides the short life cycle of insects that prevailed in the benthic community. At lower levels of biological organization, we evaluated the effects of the effluents, performing several ecotoxicological tests using selected chemicals and target species. We selected among those detected in the first study sites 11 chemicals, 3 pharmaceuticals, 3 personal care products, and 5 pesticides, and 11 target species among those present in the first study sites, selected on the basis of their relative abundance, out ecology, and also feeding habit. 5 grazers detritivorous and 6 predators. On the basis of the ecotoxicity test, we highlighted that the populations from sites more contaminated, just living downstream of the plants, are more resistant, being naturally exposed to these pollutants. In some way, they are pre-adapted to the exposure in the lab to these chemicals. And so these results confirmed, validated one of our hypotheses that the populations living downstream of the plants under natural conditions are more stressed than those living upstream. And all chemicals resulted lethal at concentrations higher than the measured ones. So suggesting that the environmental concentrations are still protective for the benthic fauna with LC50 values higher than the environmental concentration for each chemical and species. However, signs of physiological stress were detected at environmental or sublethal concentrations equal to the LC10, just considering biomarkers, so lower level of biological organization. For example, we observed a strong emission of the swimming ability in larvae. In the video, larvae of Diamesa zerniae 1 chironomid collected in the Presena stream exposed to sublethal concentrations of chlorpyrifos. Larvae were completely immobilized. This concentration was slightly higher than that one of the order of nanograms per liters detected in the Presena stream, but anyway, lower than the concentration of this compound in downstream stream reaches. And the exposure to sublethal concentrations to this compound caused also marked biochemical alterations due to oxidative stress, as lipid peroxidation and protein carbonylation, and also the accumulation of DNA damages. The comet-assay test highlighted, as we saw before with the ecotoxicity test, that the population living downstream of the plant are living in a stress condition. In fact, we found higher basal DNA damage in downstream populations. This test highlighted as all chemicals are genotoxic, at least for one population from one site. and that ibuprofen, among the others, resulted the most genotoxic. And overall, considering all our tests, ibuprofen was the most harmful chemical, and Betis alpinus, along with Betis rhodani, living more in the downstream part of the catchment, are the most sensitive species to all chemicals. So we suggest 2 new bioindicators of the presence of pharmaceuticals and personal care products in mountain freshwaters. Now I take your attention on sucralose. Sucralose is considered harmless on the basis of the previous literature, so before our project, but also on the basis of the results of our ecotoxicity test. But it resulted genotoxic for all species, more than triclocarbon and trimethoprim. So these results raise the level of concern about this compound. Our study highlighted the importance of biomarkers that must be included in biomonitoring programs. They act as early warning tools to detect early adverse effects on the health of the organisms, but they are not enough to understand the effects on ecosystems because the ecological relevance of the response increases with increasing level of biological organization. In fact, an holistic approach is necessary to environmental monitoring, as the consideration of the chronic effects on biodiversity, the cascade effects on the river food web. And it's more and more important to consider the role of other contaminants on the fate of personal care products and pharmaceuticals in rivers, like microplastics, due to absorption processes. So how to transfer this knowledge to stakeholders? So with a dialogue between scientists and knowledge users and beneficiaries through a participate process that must start at the beginning of the research project and not just in the end, and not as a one-way flow of information from scientists to stakeholders. But the knowledge transfer is more like a cycle, because the interplay between science and society is multidirectional, with scientists involved in problem identification with an assessment approach, from identification of drivers to monitoring of the traits to the evaluation of the cause-effect relationships, and from the other side, stakeholders involved in problem solving. So from the elaboration of the scientific methods along with the scientists to the development of nature-based solutions and sustainable management. The list of stakeholders you can invite on your table when you are dealing with this environmental issue is quite long. From managers of wastewater treatment plants and water supply systems to public environmental agencies, healthcare public services, enterprises, public administration, tourist agencies, and so on. On the basis of our experience, we found very interesting the organization of elicitation workshops at the beginning of the project, involving representatives and experts from each local stakeholder group listed in the box, to create a stakeholder advisory board to co-create knowledge with scientists, because the knowledge of stakeholders is a key tool to gain critical insights into current needs, to clarify desired outcomes and constraints, and to elicit the requirements on the project. We did it at the beginning of the OSTM project, inviting the managers of the local wastewater treatment plant, the local healthcare service, and also the provincial environmental agency, and we started to discuss about the need for upgrading current wastewater treatment plant. And so they recognized the importance to estimate and start to estimate the reinvestment period for advanced plants and the cost per inhabitant per year. And then the dialogue with citizens. We organized a lot of events at the museum, but also in the municipalities crossed by the river, so just in the catchment of the Noce River. a lot of initiatives, but one of the most interesting I suggest is the survey by a questionnaire that we proposed at the beginning of the project to assess the knowledge level of the local people, first of all, but as elicitation technique to highlight needs and constraints. We invited inhabitants of the valleys crossed by the Noce River, and we proposed 14 questions to our interviewers, from general questions to questions on the perception of the river quality and the problem of the emerging contaminants. The results was not bad, so people believe finally that the Noce River quality is fair, so that there are some environmental issues. And 92% of our interviewers declared that they know that the effluents of the plant are contaminated. We met our interviewers, but of course all the other inhabitants and tourists during summer in several bio-blitz that we organized just on the river, and during several public events, like several editions of the European Researchers' Night, Science Cafe at the museum, and public conferences organized just in the valleys crossed by the river. I confess you that it was not so easy to discuss some of our results with citizens in these occasions, because it is a very sensitive issue. And it's difficult to declare that some life-saving drugs that they take to survive are dangerous for the environment. From one side, we know that citizens have the right to know, especially if the research is financed by public administration and so by themselves. And from the other side, we know that we as scientists have the ethical obligation to communicate our results, but only validated information, of course, from a scientific point of view, and without creating alarmism. This is very important, without speculation based on our belief or habit. And so we decided to disseminate to citizens only data previously agreed with the local health care service. Our last message was that we can all do something, from the researchers and institutions to enterprises and citizens. Some of the actions and processes can be very time-consuming and expensive, even for the society, like, for example, the upgrading of current plants and legislation improvement, but other actions can be done immediately and are free of charge, like a more sustainable consumption of goods, do more with less, and also the adoption of more ecological and pro-environmental behaviors, like dispose of expired drugs properly, choose biodegradable personal care products, and when possible, natural drugs, and avoid waste of detergents used to clean our houses, because it's better to prevent than to cure. I exploit this occasion to thank all people and institutions involved in the different steps of the project, starting from the first brainstorming to the last publication. And of course, I thank you for your attention, and I leave you the list of the main scientific papers published to date within the RAISE-DM project if you want to learn more. And of course, my email address, if you want to contact me. Don't hesitate, please. And finally, a special thank to Professor who invited me, Valeria Lencioni. It was very interesting and a great experience for me. Thank you again for your attention.
Thank you very much, Valeria, for this really interesting presentation.
Thank you to you.